Hello, good morning, students. Last time we had studied what are nucleic acids. So we know very well that the nucleic acids are DNA and RNA. These are the two types of nucleic acids. Today we will study the structure and the composition of nucleic acids. So the first component of the nucleic acid is a nucleotide. So we know these nucleic acids, these are the polymers, these are unbranched polymers, and the repeating unit present in the nucleic acid is a nucleotide. So in short, these nucleic acids are the polymers of nucleotides. This DNA molecule consists of several million nucleotides in one molecule, and RNA molecule contains few thousand this nucleotide molecules. So in short, these nucleotides are the monomers present in nucleic acid polymer. This nucleotide consists of three components. In nucleotide, three components are present. First component is monosaccharide. It is a 5 carbon containing sugar, then nitrogen containing bases, and phosphate group. These are the three components present in nucleotide: monosaccharide, nitrogen containing base, and phosphate group. Now, the monosaccharide present in RNA is a D-ribose. D-ribose is a 5 carbon containing sugar, and the structural formula is this: first carbon contains H and OH group, second, third also contains H and OH group, fourth carbon contains hydrogen, and fifth carbon is CH2OH group. So here this OH group at first carbon, it is above, and therefore it is a beta, above the ring, and the sugar, that is a monosaccharide present in DNA, is a 2-deoxy D-ribose, that is at second position. Instead of H and OH groups, both are hydrogen group, that is OH group is replaced by hydrogen, and therefore there is a no oxygen at the second position. Hence it is called as a 2-deoxy D-ribose sugar. So remember this very well, that this RNA contains D-ribose sugar and DNA contains 2-deoxy D-ribose sugar. Then the nucleotides of both RNA and DNA contain 5-membered ring monosaccharide, that is furanodes type of structure, simply called as a sugar component. This monosaccharide unit, that is called as a sugar component. In RNA, sugar component is D-ribose, and in DNA, the sugar component is 2-deoxy D-ribose. So these components are present in RNA and DNA. Total 5 nitrogen containing bases are present in nucleic acids. 3 bases are one ring containing, that is a cytosine, uracil, and thymine. These are 3 bases. It is a one ring containing. So cytosine structure is given, then uracil and thymine. These are one ring containing 3 bases derived from pyrimidine. Therefore, these bases are called as a pyrimidine bases. The structure of pyrimidine is, it is a 6-membered ring containing nitrogen at first and third position and single and double bond in alternate position. Then in cytosine, in addition to this pyrimidine, double bond with oxygen is present at second position and NH2 group is present at fourth position. Then in uracil, this C double bond O and C double bond O, they are present at second and fourth position. Therefore, third position nitrogen becomes NH. Similarly, first position nitrogen becomes also NH. In thymine, second and fourth position have C double bond O. First and third position have NH group, and this fifth position have methyl group. This is called as a thymine. So these are one ring containing three bases are cytosine, uracil, and thymine. Then two ring containing bases. These are adenine and guanine, and they are derived from the parent compound purine. The structures of adenine and guanine, this is a purine containing this two rings. Numbering is given as a 1, 2, 3, 4, 5, 6, 7, 8, 9. This first position and third position have nitrogen, and seventh position and ninth position have again nitrogen. This ninth position have NH group in purine. Then in adenine, this NH2 group is present at carbon number 6, and in guanine, this NH2 group is present at carbon number 2. So these are the bases present in nucleic acid. Bases are mainly two types. One is pyrimidine and other is a purine bases. Pyrimidine bases are cytosine, uracil, and thymine, and purine bases are adenine and guanine. So a nucleoside, we had studied about nucleotide. Now we will study about nucleoside. This nucleoside, it is formed by joining anomeric carbon of furanose with nitrogen base. Anomeric carbon means the carbon which is carbon number 1 present in the base ribose. While numbering the atoms in the nucleoside, the primes are used for furanose numbering to distinguish from the atoms of the bases. This is given in figure 14.26. So this is 14.26. Here the numbering is given like this. This first carbon, first, second, third, fourth, and fifth, they are denoted as a prime, and the numbering, that is first prime, second prime, third prime, fourth, and fifth. And first carbon is anomeric carbon present in the ribose, and it is bonded to this base. That is a base used here is a cytosine. So this prime numbering is used in nucleoside, that is in sugar or monosaccharide component. With pyrimidine bases, nitrogen atom at the one position bonds with first prime carbon in the sugar. With purine bases, nitrogen atom at ninth position bonded with first prime carbon in the sugar. Then nucleotide are formed by adding phosphate group to the 5th prime OH position in the monosaccharide, that is a nucleoside. The nucleotides are the monophosphates of nucleoside, meaning it's in nucleoside. If phosphate group is added, that becomes the nucleotide. Abridged name of some nucleotides are AMP, means adenosine monophosphate. DAMP means deoxyribose adenosine monophosphate. UMP means uracil monophosphate. DTMP means deoxyribose thymine monophosphate. Like this, a bridge name is used. Here the first capital letter derived from corresponding base like adenosine or thymine or uracil. MP stands for monophosphate, and the small letter D, that is the beginning, that indicates the deoxyribose nucleotide. Here one example of nucleotide is given in the figure 14.27. So here AMP long form is adenosine monophosphate. So it contains this sugar component, this is the base component, and this is the phosphate. So this base present is adenosine, and this is the phosphate group present at 5th prime position. Similarly, DCMP, here D stands for D 2-deoxy, C stands for cytosine, and MP for monophosphate. So in the sugar component at second position, there is a no OH group, therefore D2 2-deoxy. Here the base present is cytosine and the phosphate group. So these are the examples of nucleotides. What is the basic difference between nucleoside and nucleotide is nucleoside do not contain phosphate group. Nucleoside contain sugar and base group, sugar and base, and nucleotide contain sugar, base, and phosphate, all three components. So that is about this components nucleotide and nucleoside. Thank you.